Welcome. It's my great honor today to introduce Professor David Kisaulis. And he just moved to the University of California, Irvine, and I think he's in the middle of moving. And I don't know if he will talk about his move in this talk, but I think he will rather talk about his really exciting work on biological inspiration, I would say, towards multifunctional materials. And David has worked in that field for many years and has a very impressive record in publications and impacts. And it is really exciting to have you today. And we're looking forward to your talk. Thank you.
Thank you very much. And Marcus, thank you for organizing this. This is a spectacular platform. So I appreciate the invitation. And thank you everyone that's joined today. I'm going to give kind of a spectrum of topics in my talk, at least three, that will kind of give you an idea of what we do. And my laboratory really has two main thrusts. Half of the laboratory, we look at organisms that are kind of extremophiles, if you will. They do something mechanically or thermally or optically interesting, and we try to understand what they're made of, how they're architected, and then utilize those designs, those architectures, but using engineered materials to mimic structures. Another half of my group asked the question, well, how do these structures actually form? What are the mechanisms of forming biological structures, biomineralized structures? And can we take cues from that, whether it be controlling pH or ionic strength or using organics to actually synthesize nanomaterials for energy and water purification applications? In the lower right, actually, these are lithium-ion battery cathode particles, all the same phase, but different shape, crystallographic orientations, just by tuning the synthesis conditions. So I'll focus much more on the first half of my group's work, but I will touch on these other areas. So the first part, I want to talk about the MURI program. This is a multi-university research initiative that's funded by the Air Force that involves a really incredible team where the focus is to try to make materials that are both strong, tough, stiff, but also at the same time lightweight. And of course, the driving force behind this is, or one of them, is to reduce emissions and of course save fuel. So you can just see here, US, we drive a lot. We don't have the infrastructure that Europeans and some other countries do around the world. But if we could reduce the weight of materials that we utilize, we could save an incredible amount of fuel, money, and then, as I mentioned, CO2 emissions. And so this MURI involves this really wonderful team. I'm really lucky to have, there's a bunch of 800-pound gorillas in the room. Mark Myers, Rob Ritchie, Horacio Espinosa are incredibly well known in the field, and they anchor this team. But there's people like Pablo Zavatieri, who's incredible and a rising star in this field. And so they are a really important part of this MURI. Cheryl Hayashi is our biologist who really informs us a lot about how these organisms got the way they are. And I'll talk about that in a minute. And then I'd also give a special acknowledgement to Joanna McKittrick, who unfortunately passed away. She was an incredible member of this MURI team and contributed greatly. And of course, the students and postdocs that really do all the heavy lifting, plus our collaborators from Air Force Research Labs, Army Research Labs, San Diego Zoo, Brookhaven, Deakin, Lawrence Berkeley, a lot of different folks that contribute to this program. And of course, the funding agencies, Air Force Office of Scientific Research and Army Research Office. And so the focus of this MURI really was to try to understand architectures in both plant and animals that provide this strong, stiff, tough type of materials while maintaining lightweight. And, biology is able to maintain this toughness even with increasing stiffness effectively by integrating and architecting organic scaffolds and then infiltrating those organic scaffolds at multiple dimensions And that will provide this extreme toughness. And not only that, there's many different components at the atomic, nano, micro scale that serve in this integration to provide this toughness, and where the structures that we're looking at have different performance metrics at different time scales as well. And I'll talk about that now. But beyond having both strong, stiff, and tough components that are lightweight, nature has been able to integrate multifunctionality into its structure. So one structure that may just, you may think it may serve a structural focus actually has additional type of functionality, whether it be sensing or self-healing, self-repair. And so we look at these organisms, and there's a number of different organisms, 8.7 million species on the planet that we can glean inspiration from. But we do ask the question, is nature actually optimized? And can we actually go further? And that's kind of what this MURI program does. It's looking at these architectures and saying, well, this organism, for example, this mantis shrimp in the lower left here, has these spear-like appendages which will impale soft-bodied prey. But those soft-bodied prey developed armor, kind of like an evolutionary arms race over time. And the spearing type of mantis shrimp could not penetrate those organisms. And it started to try and use its elbow segment of its appendage, called the dactyl club. And the ones with the larger dactyls would split off eventually to form the smashing type of mantis shrimp that we study in our laboratory. But this program says, well, could we go one step further? What would happen in the future? What would the design look like if it weren't, for example, an armored sea organism? What if it were something like this? What would the design then look like? So that's kind of the focus of this type of program. And so we look at many examples of impact-resistant, tough organisms found in different regions of the world. For example, Joanna studied these bighorn sheep where their horns are made of just keratin. So there's no mineral here, but it's the architecture that provides this incredible impact resistance. Or for example, this acorn woodpecker, watch its eye. It actually has to close its eye because the deceleration rate is so high, its eyeball will pop out of its eye when it's pecking. And then, of course, in my laboratory, we study these crustaceans called mantis shrimp that actually use these appendages called dactyl clubs to smash their prey and basically afford their ability to eat. And so this program really studies many different species in the animal and plant kingdoms, and really tries to understand if there are any common design themes that might provide this toughness and impact resistance. So I'm only going to talk about one. I don't have time to talk about all of them, and that is the mantis shrimp. And my lab, we always ask questions. And so we ask, well, what can we learn from crustaceans about making impact-resistant materials? And so the mantis shrimp gets its name because it looks like a praying mantis, and it has a shrimp-like body plan. It's a crustacean, but again, it uses these appendages called dactyl clubs to smash and accelerate through water at incredible speeds, 23 meters a second, and accelerating at over 100,000 meters per second squared, to generate incredible amounts of force. And they're only about, in our lab, we keep them, they're the Odontodactylus scalaris, it's the peacock mantis, they're about 10 centimeters long. And we keep them in acrylic tanks rather than glass, because literally they will break the glass. And in fact, they've already started to crack some of our acrylic tanks. So they impact with incredible amounts of force. This is work from Sheila Patek that showed and Roy Caldwell that shows this type of impact force from these clubs. But notice there's a secondary peak here of force that comes, and that's not from the actual contact of the club, but rather cavitation. So the organism is going so fast through this water, it's shearing the water and creating these cavitation bubbles that then subsequently implode on the poor prey. And this provides additional damage to the prey. And they can do this thousands of times. The mantis can impact thousands of times without fracturing their own club. And of course, they can afford to do this because even if they do get damaged over a long period of time, they will molt like crustaceans do and form a new club. And so we subsequently have gone in and analyzed the dactyl club, and you can see it's heavily mineralized in the CT scan. Actually, you can see the vestige from its predecessor, the spearing mantis, so it actually still has the spike on its club, but it's a heavily mineralized club. And so we've gone in and taken a closer look at this club in cross-section, and it actually consists of really three primary regions. One of our first papers that came out in Science a long time ago showed that underneath these first two layers is this what we call a periodic region, or which has this periodicity, which we found actually has this bulligand or helicoidal architecture. So these are unidirectional alpha chitin fibers that are stacked on top of each other, but subsequently rotate some small angle and form this helicoidal architecture. I won't talk too much about that today, a little bit about how we mimic that structure, but that provides an incredible amount of toughening. Above this, there's this other region, which is effectively a helicoid region, but it's compressed into a sinusoidal-like architecture or herringbone architecture. And we call this the impact region that we believe, due to this out-of-plane structure, provides a little bit more momentum during impact, but also provides a lot of impact resistance. What I will talk about is this outermost layer, which was our most recent study that we published just less than a year ago in Nature Materials, where we've looked at the outer coating of this club. And I like to make analogies. If I'm an angry teenager and I like to punch a wall because I'm angry, I may break my knuckle because of a local contact stress on my knuckle. So imagine the mantis shrimp smashing thousands of times, but it doesn't develop these high localized stresses. Why is that? And so we believe these coatings of particles coat the club and actually reduce those localized contact stresses. So we've gone in and analyzed the club, and indeed, we found that it's made of hydroxyapatite mineral. It actually has a graded structure. That impact region that has that sinusoidal structure actually has highly oriented hydroxyapatite crystals that are more or less perpendicular to the impact surface. But inside the club, where that periodic bulligand structure is, it's amorphous mineral. And you can actually see what happens upon impact. This is actually from another region of the mantis that also has a helicoid structure. It's actually its tail segment called the telson that also has a helicoid structure, but you can actually see crack deflection and crack twisting. In the club itself, we've actually sectioned the club after we didn't count. There was no one there with a clicker counting how many impacts it had, but it was mid-molt. And we basically determined by shining the electron beam or letting the electron beam sit on the sample, this secondary charge contrast imaging, that the club is actually riddled with damage, but none of the cracks have managed to actually propagate out of the club. And so again, going back to this helicoidal structure, this is this ability to localize damage is due to the ability to have crack deflection, but also crack twisting at these interfaces that provide that additional toughening. So we took that design and said, okay, well, let's take that bulligand or helicoid structure. Instead of using alpha chitin hydroxyapatite, we replaced it with carbon fiber, unidirectional carbon fiber tapes, and made helicoidal composites. And now we have a number of programs for both aerospace, wind, and automotive composites. And we've subsequently taken those helicoidal panels and performed, for example, drop tower testing on these carbon fiber composites. And what you can see is that comparing the helicoid architectures, where I have small, medium, and large angle, to what's used, for example, in the Boeing 787 quasi-isotropic, you can see the dent depth, the damage to the surface of these panels is significantly less. So what it's telling us is that damage is being spread laterally by this crack twisting versus getting through thickness damage. And in fact, after impact, the medium and large angle panels showed up to almost 20% improvement in strength after impact. The small angle didn't perform so well because the sample geometry was so small that cracks literally propagated out of the sides of the panel. So it didn't show good residual strength for that small angle. Okay, so as I mentioned, I want to focus more on this impact surface. So why don't we see stress concentrations during this impact? Here's your Bulligan structure. Here's your impact region. But at the very top of the club, roughly about 50 microns or so on the surface of the club, there are these nanoparticles. Actually, these arrows here show the pore canals that actually transport mineral during the formation of the club, but that's another story for another time. The outer surface of this club is coated with nanoparticles that are roughly about 60 nanometers in diameter, surrounded by some organic matrix. And we initially took and nano-indented the surface, the impact surface where those particles were located. And we also polished away the particles and subsequently performed the nano-indentation. So where the particles were, you see this pileup occurring, so this toughening this way. But when we remove the particles, we see cracks that are generated at these corners. So we believe this toughening is due to this, what we call mesoscale displacement of these particles. And this just shows some of these inflections here during these nano events toughening. My colleague at Purdue, Pablo Zavatieri, has subsequently just made a model showing the role of these nanoparticles so that upon impact, for example, what he's done is he's modeled the elastic modulus, where he's taken a 1-to-1 ratio. So this is the modulus ratio of organic to mineral. And what you can see that as you increase this modulus ratio, and you have these particles, you can localize these stress concentrators at the surface. And when we first published this paper in Advanced Materials 2016, we thought these isotropic particles were single crystal hydroxyapatite particles surrounded by this organic veneer. But recently we took a closer look, and what we found is that the mantis shrimp, when it first forms a newly molted structure, freshly formed, you can see it's shiny here, the impact surface is present. After thousands of impacts, that impact surface literally has worn off. And so this is, again, a mechanism to absorb energy and prevent any catastrophic damage to the underlying structure. But we took a closer look at these nanoparticles. And again, in SEM, so you actually look like raspberries, if you will, aggregated particles. But when we took a closer look in TEM, those, what we thought were single crystal particles, looked kind of porous. And the electron diffraction patterns that we subsequently have used here showed that they're not single crystal, but you can actually see arcs in these patterns, indicating that they're mesocrystalline particles. And so we've actually done some TEM, additional TM analysis of these particles. So this is just showing a few of these particles. And you can actually see that there's an interpenetrating network of organic, what we would call a bicontinuous network of organic materials that are intertwined with this, with the hydroxyapatite mineral. And within a secondary particle, a 60 nanometer particle, we actually find primary particles, single crystal domains that are stacked upon one another, but with low angle grain boundaries. And in addition, we've used AFM to show that the organic is indeed intercalated or interdispersed between the mineral phase, almost like a phase separated structure. So we see that, but we also see organic that's also occluded within the mineral. So all of these structures, or this type of architectures, we believe, provide some type of toughening, additional toughening to the club. So this is just a little schematic of what that particle might look like, where you have the mineral phase in blue, and then the organic would be dispersed throughout. And we've done some TM pico indentation on a single particle. And again, originally, when we thought this was a single crystal hydroxyapatite particle, we were surprised to see that at 80% strain, the particle would deform, and then upon release of that stress, completely recover. And so what we believe now that we know that there's organic interdispersed here is that we're probably getting some type of buckling within the particle, and of course, some type of response from the polymeric phase. And so this is a little bit more information showing this bicontinuous particle that's found in the impact surface. And again, where you can actually see some lattice distortion due to the organic that's occluded within the mineral. And so the question is, okay, this is interesting under quasi-static conditions, but what happens under high strain rate? So we have taken the club and mounted it and performed some high strain rate impact testing in this micromaterials device that we have in my lab, and impacted on both the surface as well as the underlying impact region. So the impact surface particles are removed. And you can actually see from a single impact the particle pileup that's occurring. Whereas on the right side, when we remove those particles, you can actually see these cracks developing. And this is after 100 impacts, and then also with a sharp head upon impact. And so after those impacts, we can actually see something happening. Those 60 nanometer secondary particles do exist, but we also see some remnants of smaller particles that have fractured off from those 60 nanometer particles. So there's some particle breakage here. As we actually see some fibril bridging, that's the organic phase, that may provide toughening at the nanoscale. At the what I call atomic scale, we actually see something else happening. So again, this is the intact particle before impact. After impact, you can actually see where those, I call them Lego blocks, but those mesocrystals are stacked on one another. Those low angle grain boundaries are more attuned to being fractured and will then therefore dissipate energy upon fracture. And so in addition to that, we do see some dislocations forming, as well as some amorphization during these high strain rate impacts. Again, additional sources of toughening. And this is something that our collaborator, Pablo, has managed to model. And in fact, what he's shown, for example, on the right side here, is the energy that's required to fracture a single crystal is much more high than what you would see trying to fracture something with a lower angle grain boundary. So you can actually initiate some of these fractures by having these mesocrystal-like architectures. So what are we going to do with this? So now, this is something that's work in progress. We've actually synthesizing nanoparticles and then using some organic veneers. So this is actually a silicon carbide particle with some polymer coating that we're using to basically place on top of carbon fiber composites, for example, make coatings like this to protect the underlying layer. But we're also now synthesizing mesocrystalline particles where we can tune these interfaces and actually determine, get these low angle grain boundaries where we can add additional toughening by making mesocrystalline particles. And then finally, my really talented postdoc, Wei Huang, is now making bicontinuous particles that we're now adding to these coatings and measuring toughness. But this is the direction that we're going, and hopefully in the next time we talk, you'll see me driving my Bugatti. That was my present from my PhD student. No, I'm just kidding. I asked him to to make that for me as part of his defense. But anyway, so we're looking to make coatings for helmets, automobiles, and other sporting applications. Okay, so I want to switch gears and talk about how do some of these structures actually form, and what are the mechanisms, and can we learn something from it? So there is a very large biomineral diversity in nature. I mean, many people are familiar with the nacreous layer of an abalone shell, but the incredible fidelity of reproducibility of these structures is really outstanding. And as materials chemists, we look to try to understand these processes so that we can mimic it. Brittle stars, another example of really well-architected materials using these single crystal arrays on their arms. Radiolarians just look really cool, like medieval helmets. And of course, these are sponges made of glass found off the coast of California that are actually quite mechanically robust. But how do they form? And so often, if we take a look, for example, in the nacre, it's made of aragonite. And if you looked at geologic aragonite, they typically tend to form needle-like structures. So why and how does nature control this to form hexagonal-like tablets with controlled thickness? And so if you look at this, this is just some work by Levy and Call that shows the underlying organic scaffold of beta chitin that are typically, so these are polysaccharides that are often co-associated with proteins. And those proteins contain domains that act as, if you will, sinks to enhance nucleation and create supersaturation in microenvironments. So we've subsequently, for example, this is the actual organic scaffold. It's a TM micrograph with stained beta chitin and proteins. So you can actually see the organic scaffold where those mineral tablets will actually grow. So it's this microenvironment that's, size and also chemically tuned that will control how these platelets form. And so are we trying to make, the next generation of advanced materials using abalone shell? The answer is no, we're not competing with this. We're converging these technologies. So we're trying to utilize what we've learned from biology and translate this into making nanomaterials, whether it be for lithium-ion batteries in our lab, we do this, or fuel cells or water purifying membranes. This is kind of the direction we're going. So I'd like to give an example of one organism where we study this mineralization process and we try to glean some inspiration from it. So I like to ask people, actually we're doing this virtually, but if you look at this island in the South Pacific and say, how did it form? You look at it, a lot of people say, well, it's erosion from the waves that have undercut this island to give it its mushroom shape. But in fact, the water's quite calm here. This is actually Palau. A marine biologist by the name of Heinz Lowenstamm went under this island, and what he found on the rock were these scratch marks that were underneath this island. And next to the scratch marks were these organisms called chitons. And chitons are mollusks. In fact, this is one from California, the Gumboot Chiton, Cryptochiton stellari. Chitons actually eat algae, and algae is growing on the rock and within the rock. So they've developed a specialized feeding apparatus called a radula. It's shown here in the lower left, and you can actually see its buccal cavity, its mouth, where the radular belt will come out, and it will literally scratch along the rock and drag the algae and rocky particles into its mouth, and that's how it feeds. So it has to do this in order to abrade that rock. It has to have really hard and wear-resistant teeth. So you can actually see a few of these teeth here. There's the active teeth are these last 10 rows or so. There's about 80 rows in this species that are actively used in that rasping process. And they're attached to a soft, flexible arm called the stylus. But what we find interesting is that in order to survive, what happens if one of those teeth break off? Can the organism continue to feed? So it's developed a mechanism where every few days, if you look back in its buccal cavity, you see the black teeth, which are the actively used teeth. They go to orange, yellow, and clear. The organism actually forms a new set of teeth every few days, pushes that row forward, lops off the last row of teeth, and that's how it enables its survival. But it gives us an opportunity to look and see, how does this mineralization actually occur? So the teeth themselves, just taking a cross-section through the teeth, they're tricuspid, and they have a core shell structure. The shell is actually made of magnetite, and the core is an iron phosphate called phosphosiderite. So much like human teeth with enamel and dentin, this is magnetite and phosphosiderite. And we've done some nanoindentation on these teeth, both on these transverse and longitudinal sections, and you can actually see that the stiffness is on the order of about 140 gigapascals. Hardness is around 13 gigapascals, 12 to 13 gigapascal hardness. So incredibly hard. In fact, it's the hardest and stiffest biological mineral known. So if we look at an Ashby plot, just comparing log hardness, log stiffness, and we look at tooth enamel and abalone, you can actually see where the chitin teeth lay. So incredibly hard and stiff, on par with some zirconias and aluminants, but made at room temperature. And so we've done some, I mentioned the nanoindentation, taking a closer look at these cross sections where you see the indents in the core region and the shell region. Obviously, the softer component has the larger indents. if we take a look at the shell region where the smaller indents are, we see cracks, but the cracks propagate along the periphery of the tooth. So we were curious as to why that was happening. We fractured open one of these teeth, and you can see in the shell region, the teeth actually contain nanorods of the magnetite material. And the nanorods are all oriented along the long axis of the tooth, actually providing some tensile strength, since as it's rasping, the leading edge will be under tension. But they're all oriented, and they're about 150 to 250 nanometers, depends on the location and the tooth diameter. And we took a closer look at those mineralized magnetite nanorods. You can actually see some organic fibrils that form a veneer around. Here's a region where one of the nanorods pulled out. You can actually see this organic veneer that surrounds these nanorods. And this is alpha chitin, a polysaccharide. Also, when we look closely at one of these nano indentations, you can actually see that the cracks that have formed actually go around these nanorods. So this is a cross-sectional view of one of these nano indentations. So the cracks are being deflected at these interfaces between the organic fibers and the mineral. We've also found, this is now another transverse section of this region where you'd see one nanorod, another nanorod, a third, and a fourth, where the nanorods are actually, they're not quite smooth at their interface, they're quite rough, and where they have what are called nanoasperities. And some of these nanoasperities or bumps are actually bridged between each other, one rod to another, forming a mineral bridge. And we also see a little bit of localized waviness in these nanorods. And I just put this slide up here together because we're still wondering about this. Is this a convergent solution in terms of toughening? Having, for example, nanoasperities and mineral bridges, which you also see in the abalone shell, the nacreous region, which are known as toughening mechanisms. So is this a convergent solution to toughening, or is it just a remnant of its development process, this kind of infiltration of precursor that subsequently fills out these compartments, or both? So that's just a question to kind of the community. So how do these teeth actually form? So we've gone in at the early stages of the radula and looked using synchrotron, and we find, knowing that the fully mineralized teeth are made of magnetite, early on, the organic that's present here is an alpha chitin, but the first mineral phase that we see, the orange and the yellow and orange here, is ferrihydrite. That will eventually transform to magnetite just within one or two rows of teeth. So how does that happen? So we've actually looked inside the mouth of the chitin and pulled out the entire radula. This is a little schematic showing the belt that basically will rasp over what's called an odontophore. But back in its buccal cavity, these are the active teeth that are used in rasping. But back in its mouth, the radular belt is actually folded up into a tube. And we look at the tube back in its mouth and we see this tissue, epithelial tissue, that's surrounding all the teeth. And when we remove that epithelial tissue from a surface of one of these newly growing teeth, you'll see these hexagonal cells. And within a hexagonal cell, you'll see these 100 nanometer pore-like structures that are directly adjacent to the outside of the tooth itself. And we've gone in with TEM, and we found that, so this, where my laser pointer is here, this is the outside periphery of the tooth. This is the inside of the tooth where the alpha chitin fibers are located that will actually cause this templating of mineral. But the outside is where the epithelial tissue is located and has these structures called microvilli that go adjacent to the tooth's edge. And Jeremy Shaw, who did a really wonderful study back in 2007, 2008, showed that these microvilli, so this is where the epithelial tissue would be. The tooth would be here on the right side. You can see some mineral forming. What Jeremy hypothesized was that the iron that's is being transported into the tooth. It's stored in ferritin-like structures that may undergo some, and ferritin is a ferric iron, Fe3+, that's not very soluble. We also hypothesized that maybe the ferric iron is reduced to a ferrous iron and transported through these microvilli into the teeth where they're subsequently reoxidized to form the ferrihydrite. So this is a work in progress but it's something that we're thinking about. So when you look at the first tooth, we call it tooth 0 because it's the first tooth that shows any color. We see these ferrihydrite particles, these small little domains here, forming on these alpha chitin fibrils, these long oriented fibers. And if we jump forward from tooth 0, where nucleation of mineral first forms, to tooth teeth later, you can actually see the ferrihydrite has grown significantly, but guided by these scaffolds, these organic scaffolds, these alpha chitin structures. And so this is work again with our collaborator Pablo. Those alpha chitin scaffolds are actually spaced a very specific distance apart that yield specific diameter rods on the leading edge, more narrow, that provide some tensile strength. On the trailing edge, where the tooth might undergo some compressive forces, are larger in diameter that might prevent some buckling that occurs. So those alpha chitin fibers are present in these teeth, and every time that we see a mineralized particle, it's always attached to an alpha chitin fibril. And again, it's likely in most mineralized systems, you have proteins that are co-associated with these chitin fibrils. These are nucleation sites, and it's a heterogeneous nucleation process. The particles that are nucleating here, they look, again, like a randomly oriented or organized secondary particle with primary domains within. And our initial electron diffraction suggested that. Again, looking more carefully, this is now tooth 4, where more mineral has filled in here. Looking more carefully at one particle rather than a lot of them, we see that this secondary particle does not diffract like a randomly oriented crystal, but rather, again, like the mantis shrimp, like a mesocrystal. And so how does this form? I mean, we have primary domains that are only a few nanometers in diameter that form this highly oriented structure. Is there something guiding this? And this is, again, unpublished data, but work in progress, where we found that the underlying structure to these mineralized particles actually is an organic scaffold. This just shows some amorphous regions separating two highly oriented domains within the particle. So we worked together with my former postdoc, Michiko Nemoto, who's at Okoyama, who's taken some of these proteins from extracted them from the fully mineralized teeth. And Michiko has found a number of different proteins here, including 2 that might be involved in reduction that would enable the ferrihydrite to transform to the magnetite. But she also found this one protein where we couldn't find any hits on our BLAST search, this unknown protein. We subsequently have gotten the sequence of this protein, where we've identified both a chitin binding domain shown in this orange peach structure, suggesting that this protein is bound to the chitinous fibers. But we also see this serine glycine rich region of the protein. And these serine glycine rich regions are often, via post-translational modification, phosphorylated. And then we speculated that the phosphorylation may be involved in actually nucleating and controlling nucleation of the iron oxides. And that's actually known in human teeth. Actually, there's phosphorylated proteins that actually stabilize some mineral phases. So we've done some dephosphorylation, actually showing that indeed those proteins are phosphorylated. Okay, and knowing that, we've gone back into these partially mineralized teeth here. This is tooth 4, and gone back, done some dark field analysis of one single particle. You can actually see the region where the alpha chitin will bisect this particle. But we actually see within the particle, this is EDS analysis, nitrogen and phosphorus plus iron. So again, we believe that there is this protein that is templating these mesocrystals. So what about the transformation? Again, I mentioned that the transformation occurs just within a few rows of teeth. So this is just micro XRF showing the ferrihydrite phase that exists in tooth 4 through 7, and the magnetite that starts to form at the tip of tooth 4 and propagates down and across the teeth, consuming the ferrihydrite to form the magnetite that eventually is in the fully mineralized teeth. Well, we've taken one of the teeth, I believe it was tooth 4 at the top, or tooth 5, and re-examined another particle And what we found was that same secondary particle that was originally ferrihydrite, a mesocrystalline structure, is now a mesocrystalline structure that consists of both magnetite and ferrihydrite domains. And so what we surmised is that there is a topotactic transformation between, for example, the 001 ferrihydrite plane and the magnetite 111 plane. Again, this is something that we're still examining, but it's pretty exciting. And eventually, by tooth 5 and certain regions of tooth 5, that transformation is now complete to all magnetite, and it still maintains this mesocrystalline structure. But eventually, those rods have to form, and that mesocrystalline structure will undergo Ostwald ripening. So this is just an SEM showing where the mesocrystalline part of the particle is. You can see this top part of the particle that looks quite smooth and faceted, and we can see that even more in this TM micrograph. There's Ostwald ripening of these minerals that occur that eventually will yield continuous nanorods that we see in the fully mineralized teeth. The last part I want to share, so I'm sure I'm running out of time, is how does this mineral get in here if we see something like this? So tooth 6, this is actually a cross-section of tooth 6. This is the outer region where those epithelial tissue would be and the microvilli that I mentioned that would pump mineral into the tooth. If that's true, why don't we see a higher concentration of mineral on the outside? Why do we see such a higher concentration of mineral deep within the tooth? So this was proposed by Leslie Brooker, that there may be actually two mineralized fronts, both from the epithelial tissue and maybe from this stylus, that structure that supports the tooth, this flexible arm. And you can actually see the stylus is actually a hollow tube. And actually, in terms of mechanics, during rasping, it actually serves to avoid any catastrophic failure. It supports the tooth, enables force transduction, but it actually has to be flexible as well so that it doesn't break. And it also has to be able to fold back up into the teeth. So this structure is made of alpha chitin fibers, unidirectional inside the core of the stylus. The tooth would have been here. We broke it off. And what we see is that these alpha chitin fibers continue from the stylus up into the tooth. They cross the interface, what we call the junction zone, into the tooth, and they actually support the interface between the stylus and the tooth. But they also, we believe, act as conduits for mineral transport. And we can actually see a broken off tooth here at that interface where these fibers are jutting up through the stylus into the tooth. And when we look at it in CT scan, you can actually see where the epithelial cells would be with the ferritin granules that's pumping iron into the tooth. So this top part is the tooth here. This bottom part of the stylus, here's your interface between the two. You see this plume of mineral that's occurring here, suggesting that mineral is being transported in from both directions. So where are we going with that? Well, I asked my student to make a mimic of the tooth. I said, electrospin some fibers. There's my former student, Stephen. I said, Stephen, go ahead and electrospin some fibers and let's fill that scaffold with mineral so we can make a composite. And Stephen was not successful, but we made lemonade out of lemons. So Stephen was trying to infiltrate some iron precursor around these electrospun nanofibers. But in fact, the precursor that we used infiltrated the polymer itself. And I said, well, let's do something with this. So Stephen subsequently heated these fibers under certain conditions, some reducing conditions, and the iron mineral, and now we're using nickel and cobalt as well, subsequently diffused through the polymer carbon matrix during annealing and nucleated as nanoparticles, metal nanoparticles within this carbon scaffold. So what could we do with this? And here you can actually see, for example, this is nickel, single crystal nickel nanoparticles that have nucleated within the carbon scaffold. And not only did the nickel form, but the carbon itself, we used the polyacrylonitrile, but at low temperatures, now we're finding as low as 400 or 500 degrees, we've now been able to graphitize the surrounding carbon, likely because the nickel itself, is the transition metal, is a very good catalyst for graphitization. The mechanism we're still trying to determine. But it's formed a metal graphitic type of composite material. We can subsequently oxidize the metal at a low enough temperature where the oxidation will occur. We can also keep the graphene in place, but we can burn out the disordered carbon. So now we have porous metal oxide, graphitic composite fibers. We can also remove the metal if we want, or metal oxide. So now what we're doing with this is we're implementing these structures, these composite structures, into both fuel cell catalyst, lithium-ion batteries, and also adding some photocatalytic nanomaterials where we can use this for membranes for water purification. And that's kind of the direction that we're going with this. I already thanked a lot of the collaborators, but really they did all the heavy lifting. My postdocs, grad students, undergrads, great team of undergrads, all my collaborators, including the ones from the MURI program, and then the organisms. They sacrificed the most for this, so we thank them, and of course funding from Air Force and Army. For those of you that are doing this research, it's a lot of fun. It's very hard, and it can be frustrating at times. But if you look and you study invertebrates, the good news is at the end of the day, if the experiment doesn't work out, you can always eat them. So that's me and my mom eating after a study. Thank you very much.
Thank you so much, David. That was really, really fascinating. And we have a few minutes for Q&A. So I invite you all to put your questions in the chat or in the Q&A panel, and I'll be reading them. I have one here, and I'm going to read it for you, or you can read it yourself. When you look at aragonite, what is the role of the organic phase, chitin or proteins, other than mechanical properties? Are there known genetic mutations on how they are affected? So this is something goes a little bit beyond what you've talked about, but I'd be curious to know what is known about that, and because I think everyone's always talking about the mechanical properties, but are there other features that are important for biology?
No, actually, it does touch on kind of near the last part of my talk about controlling mineralization. So if you think about it, these organic scaffolds, these polysaccharides, whether they be beta chitin in the abalone or they be alpha chitin, for example, in the mantis shrimp, one of the exciting things is these anisotropic molecules can self-assemble. And that self-assembly and understanding that self-assembly is critical in controlling, actually, and dictating where that mineral will be placed, where it nucleates, and it actually will control the crystallinity. If you think about the spacing in the abalone, and remember, these tablets are beta chitin plus some aspartic acid-rich proteins. Those localized nucleation sites within that environment, that microenvironment, will provide, again, the right conditions in which a nearly single crystal mineral can form. So beyond the mechanics, which is a really good question, we are very interested in understanding how that biotic-abiotic interface actually tunes the resulting mineralization, and not just nucleating the mineral and controlling its size, but also the crystallographic orientation. A lot of these organics have some specific stereochemistry. In fact, Joanna Eisenberg did a really cool study back in early 2000s where she took self-assembled monolayers with carboxylates and showed that by tweaking the stereochemistry, you can get different crystallographic orientations of mineral. So it's pretty exciting. And where we'd like to take that is say, let's use those organic scaffolds and maybe tuning stereochemistry or just tuning the chemistry, the chelate ability of those functional groups to actually control nucleation of non-biogenic materials. So the last thing that I mentioned with cobalt oxide and nickel oxide for fuel cells or batteries, the idea there would be, can we tune the nucleation density and subsequent size of the particles, which will then dictate the performance in the battery or fuel cell? Can we do that by tuning the pendant groups in the polymer, which is kind of, it's not mimicking nature, it's kind of being inspired from nature. So I like that question, and happy to follow up if whoever answered that would be. And my email is david.k@uci.edu.
Right, thank you. I have another one. It says two-part question. Fascinating stuff on chitons. How long would it take for the island to collapse? And the second question is a little bit more out there. Well, the first one too is how do chitons communicate with each other? And I think it's related to the question of how is there any concerted effort in the way they eat the algae and
that's a really great question. Yeah.
And then beyond that, I mean, they have to somehow communicate with each other at some point in their lives. And so I'm just curious how that would happen.
I love that question. Thank you very much. So Air Force and Army didn't give me a few thousand-year grant. I wish they did. That'd be great for stability in the lab. I don't know the answer to how long it would take to wear away those islands. That's a really interesting question. I'm not sure, but the communication question is very interesting to me. Actually, in my lab, so we have tanks in my lab where we keep chitons and we keep mantis shrimp. And if we run out of algae on our rock that we use to let them feed on, we'll just literally take scraps of seaweed and feed them. And it's interesting because I have a video and it shows the chiton sitting here, and we put the algae here, and it somehow senses that the algae is there. And I don't know enough about so apologies, I don't know enough about the biology and the sensing mechanisms within the organism, how it might actually sense that food. And it's a really good question. One thing I'll add to your comment, though, we're actually studying many different kinds of chitons, and some are found in Japan, Australia, Mexico, US. There's many different species. And one of the things, it kind of goes back to your first question, there's actually different morphologies of those teeth. You can actually have, there's the cryptochiton has tricuspid teeth shapes, and the algae that it eats is tall and grass-like. So we actually make this analogy almost like to a garden rake, where it's just raking in the food. So it doesn't really maybe abrade away the rock as deeply as other chitons do. So the one we study in Mexico actually has a monocusp. And it's so the morphology of the tooth is different. The materials are different, and its length of its stylus is actually shorter. But we find that eats more crusted. It's called coralline algae. That's much harder, and we actually see those teeth get worn away a lot easier. So it's very interesting in terms of structure, function analysis, as well as the environment that they live in. But we do see rock being abraded away by those chitons at a greater rate. So it's very interesting.
Yes, fascinating. I never heard of this other than in your work. There's another question and about the eyes of the mantis shrimp. I mean, they're very famous, right? And the question is, are you familiar or have you looked into any interesting mechanical phenomena related to the eyes? I mean, it's a very interesting optical system, obviously, but and very impressive. It looks impressive, and it has, I think, some very interesting, receptor dimensionalities. But there could be some interesting mechanics, perhaps. So the question is, what do you know about that? Excellent
question. And actually, we haven't studied the mechanical properties of the eye, although my specialist, Wen Yang, is actually looking at how these eyes can pivot, right, on their stalk, which is quite interesting because they can really move in many different directions. That's interesting. Yeah. In terms of the mechanics, people have studied, for example, the eyes of trilobites. These ancient organisms where they believe that they showed that the eye actually was, the lens was mineralized. I think, and so the question here is, I don't know, I think the mantis eyes, I don't think they're mineralized, but there is a group also, actually they're part of the Air Force research labs that are also studying these eyes and these lenses and their subsequent mechanical properties. And again, for the person who asked that question, happy to even put you in touch with those people that do that work. But we are now, those same coatings that I showed here, these are, this is just a urethane coating with some ceramic in it. But we're actually now looking at transparent coatings exactly for optical systems that are both robust mechanically, but also optically transparent.
That's great. Well, fascinating stuff. So thank you very much again. I think we're at the top of the hour now. So thank you, David, for really an inspiring talk. And we have enjoyed also, especially the discussion. Thank you all for the questions. And with that, we're concluding today's JMBBM Frontiers webinar, and we hope to see you at the next one in a couple of weeks. Thank you.
Thank you, Marcus. Thank you, everyone.